Hello again. We are back and in action, and we're going to talk today about immunohistochemistry, or IHC. So this is a method that we're going to use to be able to detect very specific types of proteins. So we've done the staining methods where we sort of just do general dyes that get bound to either acidic or basic residues within within a given tissue section. So we can get basic cellular staining. We can see a lot of structure. We can differentiate between white matter and gray matter of the brain. And to some degree, we can sort of look at a little bit of morphology of cells. And all those things are wonderful, right? But sometimes we actually want to be able to quantify the amount of a specific protein. Or we might want to be able to tell if a certain type of cell is making a particular type of protein. Or we might want to see if a treatment induces the production of a certain type of protein. So there's lots of different reasons why we might want to do this, okay? And in order to visualize only one specific type of protein, okay, in contrast to something like H&E staining where you're going to be essentially using a dye like eosin that stains all the protein that's there, okay? To be able to do it specifically, we have to use a method called immunohistochemistry, okay? So here, as I said, we're going to be detecting specific proteins in tissues, okay? And the way that we do this is by exploiting a tried and tested method of antigen recognition, that of the adaptive immune system. So if you know something about the immune system, you know that one of the ways in which we prime our bodies to be able to fight an infection that we've seen, okay, in the future is by making molecules, right? Big, actually quite large molecules with lots of 3D structure, and they're called antibodies. And antibodies are produced in response to specific types of what we call an antigen. So an antigen is a particular sequence that an antibody is going to bind, okay? It's going to bind to. So typically an antigen is part of a protein. So it might be part of the amino acid sequence is going to be recognized by the antibody, okay? You remember that proteins are made of amino acids and all of these things. So in your body, for example, you get strep throat or you get the flu, and your body sees these what we call pathogens, so these nasties, right, these microbes, and they contain certain proteins on their exteriors. And once your body sees these, we have very specific types of immune cells that will record this information. And we actually have cells called B cells and plasma cells, all right, which are specific types of B cells. These plasma cells will start making lots of antibodies, okay, that will recognize that specific sequence, okay, of the antigen, of the invading organism. Now, why is this important? Because we can actually prime a system to recognize a certain type of protein, okay? Our bodies do this to defend us from that same flu or cold if we see it later, okay? And we're going to use antibodies to actually be able to bind to specific types of proteins that we want to see, okay? So antigens are the proteins of interest, okay? That's what we're going to be calling them in this particular type of method, okay? Once we actually bind them, we have to be able to see them. So we're going to be using antibodies that binds what we call selectively, okay, to a specific antigen. So what that means is that I'm going to have an antibody that was generated against a certain antigen. Say it's generated against a HIF-1 alpha protein. That doesn't matter what that is, okay? I could have said protein X, right? But if I have an antibody that's generated against that particular protein, it should only recognize that protein and nothing else. That's the only thing it should bind to. And so, if we then attach something to the antibody after it has sort of bound that antigen, okay, we can attach something else that has either a colored or, a fluorescent molecule attached to it or an enzyme attached to it, okay? And what these things are going to do is they're going to allow us to actually see color in the location where that protein is, right? And it's sort of like building a scaffold, and we're going to show you that in just a couple of seconds, okay? A couple important things to know, though, all right? Whenever you use IHC, there's a couple things we have to do because antibodies are sticky. And even though you think that you're going after only your protein and you're only binding the protein that you're looking for, you might in actuality be binding your antibody to other things in the tissue, other types of proteins, okay, without knowing it, because methods aren't perfect and there's lots of reasons why this could happen. So, with that in mind, what we have to do is always validate this methodology. And so what we always need to make sure that we do, as we would with any experiment, but in IHC it's particularly important, is to use both positive and negative controls, okay, to make sure that we're confident that we're actually only labeling one protein. And then using additional assays, right, or additional experimental methods is always advisable to validate your findings from one. Okay, so let's get to it. This is the IHC method. I'm going to kind of walk you through this, okay? So what we're dealing with is we're going to be starting with a tissue. Now that tissue is made of cells, right? And these cells make proteins. So let's say that in our particular case, we're going to be trying to find protein X, okay? So we want to measure the total amount of protein X, or we want to see what cells are expressing protein X, okay? Could be either or. So here's our cell or our tissue. Here's the proteins, okay, seated on the membrane of these tissues. Now we can actually use IHC to detect proteins inside a cell, or on what we call what we actually refer to as cell surface antigens, which are proteins that are bound to the cell membrane and sort of stick out into the extracellular space. Just depends on what you're trying to do. All right. So we're gonna have cells, okay, or a matrix. So remember, that space in between all the cells in a tissue is what we call extracellular matrix. So sometimes, right, we're not actually interested in production of a protein by a specific type of cell, but we might want to know how the extracellular matrix is being remodeled, okay, maybe after an injury or maybe after a treatment. So there's proteins there too. So we might want to label something that's in the extracellular matrix. But in either case, okay, the first thing we're going to do is we're going to incubate these sections with what we call a primary antibody. All right. Now the primary antibody is the antibody that's actually going to bind to the protein that you're interested in. Okay, what we are going to call our antigen, right? The antigen or the protein of interest. So here you can see this primary antibody. We're going to put it in here. It's going to stick to this protein. It's going to bind, okay, to this protein. Now, this antibody was made so that, in theory, it should only recognize this protein, because this protein has a certain amino acid sequence on it that this antibody remembers, okay? And whenever it sees that amino acid sequence, it's going to bind to it, okay? So that's how this works. So we have the primary antibody is going to bind first, okay? Then we're going to put on a secondary antibody. Now there's a couple different ways to do this. I'm going to show you one way, okay? And this is actually with this sort of scaffold method, we're going to put a secondary antibody on. Now the job of the secondary antibody is to stick to the primary antibody, okay? So the secondary antibody is going to bind the primary antibody. So it's going to bind the antibody that's actually attached to our antigen, okay? It's going to make it a little bit more prominent, okay? A little bit easier for us to see it. And it's going to have something attached to it or conjugated to it. We call this conjugated, okay? So secondary antibodies tend to be conjugated, all right? And there's two different ways that this can happen, all right? We can either visualize our protein of interest by a precipitate that's formed from an enzymatic reaction, or we can actually use, we can detect direct wavelengths, okay, that are emitted from a fluorescent fluorophore, or a fluorescent molecule, okay? So these are the two that are shown here. So on the left, you can see we have a secondary antibody, and it actually has HPRO, okay? So this is actually horseradish peroxidase. This is an enzyme, okay? And when we incubate these tissues that have all of these antibodies bound to them, okay, with something called DAB, this is a substrate for this enzyme. And when these two come into contact, a colored precipitate is going to form. And that's what we're going to visualize, right? So this precipitate will only form in places where our protein of interest is present. So this is going to actually allow us to visualize where this protein is in the tissue section. Make sense? Likewise, we could do a similar sort of thing, but instead of using an enzymatic reaction, we're going to use an immunofluorescent. Okay, so here we actually have a primary antibody, and they have omitted the secondary antibody altogether. So we can actually conjugate a fluorescent tag directly to a primary antibody, or we can add a secondary antibody that has the conjugate on it. It can be done both ways. There's pros and cons. It just kind of depends. All right. But that fluorescent tag is going to fluoresce at a certain wavelength. Okay. So when you use a high-powered, super fancy microscope that has fluorescent filters in it, and you're looking at a specific wavelength, you can actually see the fluorescent signal emitted. Right. Okay. So what do we need to know about this? What's so important about it, okay? As I already said, you have to have controls. Positive and negative controls are critically important. Also important to always wash sections very thoroughly between steps. So we're going to be starting out rinsing tissues, cleaning them off, essentially vanquishing them, we hope, of all extraneous salts or other things that might be stuck in the tissue. We're going to incubate them with our primary antibody. Now, this antibody should bind only to the protein of interest and nothing else, right? But once we put this solution on there that has the antibody, and it's going to diffuse in normal ways, right? From high to low concentration, it's going to start to bind. There might be a bunch of antibody that doesn't bind anywhere that's still laying around in the tissue. And what we want to do is make sure that we wash all that off, because we don't want any reaction products floating around in places where they're not actually stuck to proteins, okay? And this can happen. So we want to make sure that we eliminate what we call non-specific binding, okay? That's basically false positive signal. So it looks like there's signal, but it's not actually real, because for whatever reason, things might have stuck to something else in the tissue, and we don't want that to happen, all right? The other critical component of using IHC, and this is maybe one of the most important things, is making sure that we select the appropriate method for detection. So, for example, HRP, horseradish peroxidase, okay? If we're doing immunohistochemistry in a liver, for example, now, we have in the body organs that we call organs of hyperfusion, so that means that they actually receive a great deal of blood supply. Now, the liver has a lot of blood, okay, that is really trapped up in it. And anyone who's ever removed a liver sample knows that it's a very sort of very rich in red blood cells, okay? Now, one thing that red blood cells and blood has in it is a lot of peroxidase, okay? We call it endogenous peroxidase because it's endogenous, meaning it's contained within the body. It's not something we add, right? But it's peroxidase, okay? Now, horseradish peroxidase is actually an enzyme that's going to react with this, okay? So what we don't want is our HRP reacting all over the liver tissue, okay? Non-specifically, just because there's a lot of blood in there. So we might choose an antibody for doing immunohistochemistry in a liver sample that does not utilize the HRP enzyme, okay? Because we know that if we use this HRP enzyme, we might end up getting a lot of non-specific reactivity because there's going to be lots of peroxidase actions going on in there, okay? So we might use something else. Maybe we'll use something fluorescent, or maybe we'll use biotin. There's lots and lots of different types of conjugates. You guys don't need to know all those things, but that's just something to consider. And then, of course, we're going to select the right kind of antibodies. So it's worthwhile to kind of go through the process because the terminology can be a bit confusing, all right? So how does an antibody actually made and how do we pick it, all right? Well, the first thing that happens is you actually have an organism, and it can be any organism, okay? Here, there's a mouse shown in this diagram here, all right? But it could be anything, and they're going to be injected with an antigen. Now, just like happens in all animals, really, in us, for sure, right? You get exposed to some sort of antigen, okay? And what's going to happen is the adaptive immune response is going to kickstart, and you're actually going to start producing antibodies against this antigen, okay? Because it knows, your body knows that this is something that does not belong. It is a non-self molecule. And your body recognizes that it's non-self and it doesn't belong there, and it's going to create antibodies against it, okay? So if I took a piece of a protein, okay, from a guinea pig, and the protein is different than the one that this mouse makes, and I inject that piece of the guinea pig protein, into the mouse is going to recognize that that protein is not supposed to be there. It's non-self, and the mouse is going to mount antibodies against it. Okay, so when that happens, okay, what then occurs is some time is given for that animal to actually mount the immune response to start producing lots of antibodies. So those B cells, okay, they differentiate into a specific type of antibody making cell called a plasma cell. All right, so the plasma cells are the ones that make lots of antibodies, and they're going to make a bunch of antibody. And then serum, okay, so blood, one component of blood is serum. We're going to collect this from the animal, and it's going to contain all the antibody in it. Okay, and it's going to be purified so that we're going to take the serum and we're going to get rid of everything else except for that antibody, okay, that's generated against that antigen. And there's ways of doing this. We're not going to get into the details of how they do it, but essentially they can use columns that they know will stick to a certain part of the antibody and only that antibody, right? So they can purify them out. From this point, all right, we can either use that fraction, okay, that we've just collected. So this is the actual antibody from the serum. Or we can actually do a second sort of method. Now this second method is what we call producing a hybridoma line. All right, and what a hybridoma line is essentially we take those plasma cells, those antibody forming cells, and we fuse them with immortalized cell lines. Okay, so these are basically tumor cells. They're cancerous cells. Okay, so they're just going to do what? What does cancer do? It divides and divides and divides and divides and divides. So by fusing these antibody producing cells with the tumor cells, we're going to create these cells that are going to divide and divide and divide and divide and keep producing and producing and producing and producing these antibodies. Okay, so and then what we're going to do is we're going to individually select out individual clones of these. So what we end up with are lots and lots of antibody production, okay, of a very specific antibody, okay, and that's what we call monoclonal. All right, so hybridomas can be used. A lot of times they don't use hybridomas, and the difference really is sort of how specific you want to be and how much detection you want to get. And we'll get to that in a couple of seconds, okay? But I am pointing out here, and the reason why I went through the whole purpose of doing this is because the language of antibodies can be confusing. For example, if I say that something is a goat anti-rabbit, you probably have no idea what I'm talking about. In a couple of minutes you will. So just one thing to keep in mind as well as you're watching this is that I tend to write AB as my abbreviation for antibody, just because it's simpler than writing it out every time. It's somewhat of a standard abbreviation in the field. So that's antibody language there. All right, so what is the nomenclature? Okay, we're going to use an example. So in this example, the goal of my experiment is I want to detect a protein called myelin basic protein, okay? Now this is one of the key proteins that makes up myelin, right? The myelin sheath that myelinates neurons. And I'm going to do this in a pig after I've treated it with a new drug that I think is really going to help to cure ALS. So ALS, amyloid lateral sclerosis, is actually a demyelinating disorder, and so you might expect that if somebody has a demyelinating disorder, if their myelin is being destroyed in their body, you will see changes in one of the major proteins that makes up myelin, right? So if we give a treatment, we might expect that there might be more or less myelin basic protein around depending on whether or not the treatment's working. So let's say we're going to use IHC because we've given a drug in an experimental model and we want to see how much myelin is left. Is the treatment working? Okay. So the first thing we're going to do is we're either going to make, okay, so generate on our own by injecting animals and all these things, or purchase a primary antibody, okay, that is made against the myelin basic protein that is in the pig, okay? So that species, their myelin basic protein, is the one we want to detect, right? Because remember, our model's in the pig. Most people will purchase the antibody. There's custom labs that make these. It's a very expensive and long, arduous process to make your own antibody. I've done this before, and it's not fun. And most people would much rather purchase them for a few $100. Then you gotta just sort of make sure that you're getting a good one because that can be a problem too. But so the first thing we're going to do is we're going to start with the primary. Now the primary antibody, remember that's the first one that we're going to put down, is the one that's going to recognize the pig's form of this protein, okay, the myelin basic protein. All right. Now remember, to have an antibody against the pig myelin basic protein means that some animal, okay, must have been injected with a piece of this protein and knew that it was from a pig and not from itself. And it was able to create antibodies against it. So that means that we could have used many different species to actually make this antibody, right? If we're using a piece of the pig MBP because we want antibodies against it, we could have injected this piece into a mouse that would have made antibodies against it, into a rabbit that would have made antibodies, into a donkey, etc., right? Into a sheep, anything. So it's important to recognize that, okay? Because that antibody that you have against myelin basic protein that's going to recognize the pig form was made in a certain species. So what you're going to end up with, okay, is something like anti-MBP rabbit polyclonal, for example. All right, polyclonal just means basically that it was not a hybridoma cell line. Okay, so it actually can recognize multiple different sites on the protein instead of only just one in the case of the monoclonal. But I'll go over that in a minute too. So but the idea here is this is a rabbit, okay? So this means that it is anti-MBP. Since we looked in this case for the pig form, okay, it's going to recognize the pig form, but it is a rabbit antibody because it was actually made in a rabbit, okay? It's a rabbit immunoglobulin, okay, that actually will recognize the pig form of myelin basic protein, okay? Or you might see it written as rabbit anti-MBP, all right? So that's the primary. So when we use the secondary, we have to use a secondary antibody that's going to bind to the primary antibody. It's just like a scaffold, okay? So the secondary antibody, okay, is named for the species that the primary antibody was actually generated in, okay? So in this case, right, if our primary was made in a rabbit, then our secondary antibody would have to be anti-rabbit, okay? So that's actually going to recognize the rabbit antibody, right? Because remember, if we're in a pig, there shouldn't be any other rabbit antibodies around except for the one that we've incubated the tissue with, right? I mean, we don't have a mouse making sheep antibodies under normal circumstances, okay? The only way that a rabbit antibody could have gotten in to this tissue is by us actually putting it in there. Okay, so anywhere that there's rabbit in the tissue should be the one that we've just added because it's actually a pig, and the pig doesn't make rabbit. So now we're going to use an anti-rabbit to bind that one, okay? And you might see it written a bunch of different ways. Like, for example, it could say Alexaflor 488 goat anti-rabbit. Sounds confusing, right? What this means is that it is anti-rabbit, so it's going to recognize a rabbit antibody, okay? The goat part means that this one was made in a goat, okay? So what they did now is they took a goat and they injected it with rabbit immunoglobulins, okay? So any type of antibody, doesn't matter what it is, any rabbit immunoglobulin, this thing is going to recognize, including the one that we made against myelin basic protein. You get it? Okay? Alexaflor is just the conjugate that's on the secondary. So this one happens to be a fluorescent molecule. Okay, Alexaflor 488, because it's excited within the 488 nanometer range, okay? You don't really have to worry about that. We're not going to be using these probably here, but it's just something to note. So this is the conjugate, and this is actually, over here, goat anti-rabbit tells us it's generated against the rabbit antibody, but it just happened to have been made in a goat, okay? Or you might see goat anti-rabbit alkaline phosphatase. So what do you think that this means? Well, we know whenever we have ACE, we have an enzyme, right? So this antibody, unlike the Alexaflor, which was a fluorescent molecule, this one was conjugated with an enzyme, okay? So what we're going to do at the end is we're going to add a substrate that reacts, okay, with alkaline phosphatase to produce a color that we can see in the tissue. Make sense? Okay? So I understand this is confusing. This is confusing to just about everybody who does this. If you think that you've got it, then you're awesome. If you don't worry about it because we're going to talk more about it in class too. So, but it's just something that I kind of want you guys to get exposed to now before we actually do the method. Because when we start working with the reagents and the antibodies, this will make a little bit more sense, all right, when the time comes. Okay, so this is just a slide that I grabbed actually from. This is a good one because it kind of shows the difference between a polyclonal and a monoclonal antibody. All right, so there's advantages and disadvantages to each, but really I just want to call your attention to a couple of quick things about it. Okay, number one, okay, you can see here on the polyclonal we have AB1, AB2, there it is, AB2, AB3, and AB4. Now these are four slightly different sites on the same antigen. Okay, so what that means is that this antibody that this mouse in this case actually produced all on its own, okay, just from seeing the non-self antigen, it actually produced four slightly different antibodies, okay? Now they all recognize the same antigen and only that antigen, but they recognize slightly different sequences on it, which means that we actually have more of a chance of actually recognizing the antigen in the tissue because we've got four different shots at it here, four slightly different sequences. Whereas the monoclonals that we make, okay, each clone is selected, each individual colony, and it is only has one sequence, okay, or sometimes we call that an epitope, okay? It only has one epitope or sequence that it recognizes, okay? So if we have one given monoclonal, okay, antibody, it came from one immortalized cell line, okay? Every single cell was exactly the same, and they only recognized one single site on the antigen, okay? So pros and cons. Well, if you really want to get signal and you want to try to get the most bang for your buck, usually the polyclonal is the way to go, right? You can see just from the image, basically you got four different, slightly different epitopes that you can see, so you're probably going to increase your likelihood of seeing it, okay? The drawback to that is you might actually get a little bit more non-selective binding, okay? It might not be as clean. You might get a little bit more background, a little bit more non-specificity. Whereas with your monoclonal, since it only recognizes one and only one specific site on the antigen, right? Your monoclonal is going to be very selective, very specific. So you should, in theory, get less background, right? Less non-specific binding, but you might also get a weaker signal. And you may miss some of the effect if you're not actually capturing all the antigen, okay? So there's always a trade-off between these. And really, there's other things I could talk about. I'm not really going to. Those are a couple of the big ones that you'll typically grapple with if you're doing these kinds of methods, right? I mean, realistically. And the other slide here, and this is kind of just the summary of that, but this one down here just shows different antibody modifications, okay? So these are different conjugates. So here you can see a fluorophore. That's basically a fluorophore is a fluorescent molecule, okay? So this antibody has a fluorescent molecule attached to it that you're going to be able to see fluorescence, so you'll be able to visualize where it is. Whereas this one here, okay, and we used actually both of these. AP stands for alkaline phosphatase, which is one I talked about. This is horseradish peroxidase, which is one we talked about too, okay? So this actually has the enzyme conjugated to it. And there's a couple other things here too. Beads a lot of times are used to actually purify them. They use bead columns. Biotin streptavidin is just another system that you can use to visualize these, okay? So it's the same exact concept. We don't really need to get into that too much. Prelude to Western blotting, for now, we're going to leave this alone. When we actually do our Westerns, we're going to go back to this slide and we're going to talk about it a little bit more. But for now, essentially what we're going to be doing is looking in tissue sections to see if we can detect injury markers, okay? And that's what we're going to be doing. So I hope you found this helpful and intriguing and awesome. And here's a little quiz that you can take to see how well you took in all this information. And the next time we chat with each other, well, I chat to you, I guess, I'll be talking about the different cells of the nervous system, okay, neural cells, and which is going to include neurons and glia. All right, thanks.